On this week's Arabidopsis Research Roundup, I'm absolutely delighted to be joined by Dr. Stephen Spoel from the University of Edinburgh, who is also the chair of the GARNet Advisory Board at the moment, so we've been working together over the last couple of years. And we're talking about a paper just published in PNAS, the title of which is Nucleoredoxin Guards Against Oxidative Stress by Protecting Antioxidant Enzymes. So this is a really interesting story, and the lead author on this, Sophie, she came to the GARNet 2016 meeting last year and had a poster on this, so it's great to see that it's gone from poster and actually has now been published. So thanks very much, Stephen, for joining us. And it would be great if you could give us an overview of a bit of background to the paper and then the main findings. That would be great.
Yeah, sure. Thank you very much, Greg. So in my lab, we're very interested in reactive oxygen species. And reactive oxygen species are actually quite a damaging agent to any kind of cell. And the cell has learned to harness these molecules, both to protect itself against those molecules, but also to harness them as signaling molecules within the cell. So we would like to understand in my lab how cells protect themselves and how they actually use these really reactive oxygen species as a controlled signaling switch in the cell. And that's kind of the what set this whole story into motion. So in my lab, we've been looking at a lot of enzymes that are antioxidant enzymes. Everybody's probably heard of antioxidants. Their role is to detoxify these reactive oxygen species. And this becomes particularly important upon stress. So things such as pathogen stress or abiotic stresses often lead to an enhanced increase in reactive oxygen species to a level where it actually becomes dangerous to the cell membrane, to DNA damage, etc. And so antioxidants then come in. They're often upregulated by the cell to detoxify these reactive oxygen species. So they're very important agents within the cell during all sorts of stress responses. So in this story, we looked at one such enzyme called nucleoredoxin. And nucleoredoxin mutants show an enhanced resistance response, showing that they probably have a perturbation in their intracellular redox status. In other words, they probably have different levels of reactive oxygen species within the cell. So we looked at this a little bit further, and I think the main sort of finding that we had here in this paper is that we looked for targets that this enzyme might have, so protein targets. And surprisingly, we came up with yet another set of antioxidant enzymes that was being protected by this nucleoredoxin. And in particularly, we studied here the role of catalase. And catalase is an enzyme that directly detoxifies hydrogen peroxide. Hydrogen peroxide is a very reactive oxygen species. And what we found is that these antioxidant enzymes, such as catalase, themselves, of course, function in an environment where there are lots of reactive oxygen species, such as hydrogen peroxide, and somehow they don't get damaged by these agents. And we discovered in this story that they actually do become damaged, but they are protected by the action of nucleoredoxin. So if you take nucleoredoxin away, the function of catalase and probably other antioxidant enzymes goes down.
Okay. So can you give a little description how, or do you know how, the nucleoredoxin is able to protect the catalase enzymes?
Yeah, so catalase contains several different cysteine residues, and cysteine residues are very susceptible to modification by reactive oxygen species. And their modification can lead to conformational changes, or if there's an active site cysteine present in an enzyme, this cysteine may be blocked by reactive oxygen species modification. So we think that this nucleoredoxin ensures that these cysteines stay in the reduced state and thereby protects the activity. Whether this is through active site cysteines in catalase, or whether this is through protecting any conformational or charge change of catalase, we do not know yet. But it's clear that cysteines, clear from our work, that cysteines are kept in a reduced state in catalase.
Okay, and so, I mean, at the heart of this paper is a kind of a mass spec screen that you did, where you used a modified form of the nucleoredoxin, kind of to trap partners. So can you describe that screen that you used initially?
Yeah, sure. So we use recombinant nucleoredoxin protein, and nucleoredoxin is part of a wide family or larger superfamily of thyrodoxin proteins.
Okay.
And thyrodoxins typically have two cysteines in their active site, and they use these cysteines to attack other cysteines on other proteins that are in an oxidized state. So the first cysteine in the active site of nucleoredoxins, or thyrodoxins in general, attacks the modified cysteine of the substrate, and that essentially creates a link between the two molecules, so between substrate and nucleoredoxin. And this link is covalent, so it's incredibly stable. The second cysteine in nucleoredoxin active site is then required to resolve this link, so that the substrate is left reduced and the nucleoredoxin is left in the oxidized state, and the nucleoredoxin can then be recycled for its activity. So if you mutate the second cysteine in the active site, you eliminate the ability of nucleoredoxin to resolve this link with its substrate, and so you can covalently start trapping substrate onto your recombinant protein. And so that's the strategy we used. So we then took mutant nucleoredoxin and wild-type nucleoredoxin and compared the substrate that we trapped to see which were the true substrates using a proteomic analysis.
Okay, cool. And I urge listeners to have a look at the paper and show that it was a really nice set of enzymes come out of that analysis, and it's nice when you do the experiments and you get a load of stuff which you think makes a lot of sense moving forward. Certainly. So
so in this case, of course, we didn't only catch catalase. We got a whole set of different enzymes, and I think one of the most striking things is that we appear to have caught most of the antioxidant pathway involved in detoxification of hydrogen peroxide. So we caught catalase. Notably, we caught all three catalases within the Arabidopsis genome. We also caught ascorbate peroxidase, which is another important direct detoxifier of hydrogen peroxide, and several enzymes that support the activity of ascorbate peroxidase. So there appears to be the whole hydrogen peroxide detoxification pathway that we picked up here, which was quite special.
And so would I be right in thinking, for some of your future work, you might be looking at the functional relationship between the nucleoredoxin and those other peroxidases as well?
Absolutely. There's two things I think that are very interesting for future work. One is indeed looking at other substrates to see, well, what else is nucleoredoxin protecting? This is important because we might not only have hydrogen peroxide as a detoxification pathway that's being targeted, but maybe other reactive oxygen species as well. And the second thing, which is of interest, I think, to us, is that the superfamily of thyrodoxins to which nucleoredoxin belongs is quite large. And particularly in plants, it's a highly expanded family. And so understanding the individual contributions of each of these different dozens and dozens of enzymes in the plant genome would be, I think, a great step forward in understanding redox and understanding detoxification of reactive oxygen species.
Absolutely, considering, especially since this one protein seems to have quite a major role. So just as a final question, if we can draw out a little bit and think about the bigger picture, perhaps. So in the very first figure on the paper, you show that the nucleoredoxin mutants have an enhanced disease resistance. So how does this linkage and on the kind of biochemical level, how does this speak to a larger impact about crop health or other things of that nature moving forward?
Yeah, of course. Nucleoredoxin itself indeed has a higher pathogen resistance. I'm not sure if this is a correct strategy to follow, looking for a crop protection approach. The reason for that is because disruption of nucleoredoxin leads to some form of oxidative distress within the cell. Okay. That's not an environment pathogens like. But if it's too much, this can also lead to changes in growth, of course, and that's undesired. However, a strategy that has already been followed in agriculture is to take certain antioxidant enzymes, such as catalase, and overexpress them, essentially to catch away any reactive oxygen species, particularly in conditions of stress. So I think our work now shows that perhaps expressing only something like catalase is not enough. You need to probably co-express something like a nucleoredoxin to ensure that this extra amount of catalase is truly active and doesn't get inactivated by the sheer amount of reactive oxygen species that accumulate upon stress.
So a final question, maybe slightly tongue-in-cheek question just to finish. So, you showed that the nucleoredoxin protects the catalase in this difficult environment, but then you go back one step and you think about who watches the watchman, what protects the nucleoredoxin in that environment as well in the cell. Have you got any idea about that or strategies?
Yeah. So of course we don't really know what protects a nucleoredoxin. But I think one key thing about the superfamily of thyrodoxins that nucleoredoxin belongs to is that their cysteines are usually only exposed in the active site. And because the active site cysteines interact with each other and resolve each other, any modification of one cysteine can be undone by the other cysteine. So this is, I think, an evolutionary trick which prevents any thyrodoxin enzyme from becoming modified in a damaging way. That's speculation, I think, of how it's fully protected, because, of course, other residues may also be modified by reactive oxygen species. But I think that's probably one of the main reasons why thyrodoxins and nucleoredoxin can function in such environments.
Yeah, I think that sounds like a reasonable starting hypothesis, I think. So, okay, thank you very much, Stephen, for talking to us today. And as ever, we look forward to hearing about the future work in this area from your lab. Thanks.
Thank you very much, Guy.